So, good afternoon, everyone. Welcome to week 2 live interaction session for the course Classics in Neuroscience. So, is my voice audible? Is my voice clearly audible?
Yes, ma'am.
And is the screen visible?
Yes, ma'am.
Okay. So, in case you have any doubts, I can start with addressing those first. Otherwise, we can discuss the previous year question answers, assignment questions and answers, and discuss some of the key concepts that were covered in this week's lectures. So, does any one of you have any doubts that you would like me to first address, or shall I start with the Q&A?
Ma'am, I just joined the course, so I don't have any doubts as of now.
Okay, so do you want me? So, shall I first go over the concepts of the week and then we can discuss the questions? Is that okay?
Yes, ma'am. That will be
Oh,
helpful. Thank you.
okay. So, we first start with so in the beginning of the 19th century, people were trying to understand how the nervous system actually develops. So, how does it form? So, just like every other cell of the body, the cells of the nervous system also develop from the embryo. So initially at that time, the technology was not very advanced. People did not know how to people did not have the tools and techniques that we have right now. So what were they doing is Wilhelm His was the one. He's a German who lived in who had his laboratory at Basel, Switzerland. So he's the one who first started looking at the embryo. So he thought, okay, let me see what how the nervous system is developing by looking at different. So he used human embryos of different ages. He collected these embryos over several years, human embryos of different ages, and he stained that. So he stained that and he observed them under the microscope and he tried to see that what is the sequence of events. Well, how is the nervous system actually developing? So what he did was when he used these embryos of different ages, he saw that there is from the spinal cord there is one long axon that is being formed, and from that axon there are multiple shorter processes that are forming. So that is so he used this to understand the sequence of events of the development of the nervous system. And that is true even now. So even now it is just a minute. So this historical tradition that he started, that is using observations of fixed and stained tissues, you're trying to understand the mechanism. So this is called historical tradition in developmental biology, and he pioneered that. So he was the founder of neuroembryology, and he made this very important observation that first the a developing motor neuron will first send us long axon, and from that there are multiple short processes that emerged. And so from that there are two rules that have come up. So the rules are that nerve cells always produce only one axon, and the second is that the axon develops first and the short processes develop later. So initially, other people were calling these short processes as protoplasmic extensions. He changed the name to dendrites. And that term has been adopted even now. So even now, people call these short processes as dendrites. So William His, who was the founder of neuroembryology, he introduced the term dendrites, and he is the one who, sorry, just a minute. Okay. So he is the one who pioneered the use of embryos, because right now this seems very common to us that, oh, we can use embryos to study these development, we can study nervous system development also like this. But at that time very less people were actually using embryos to study these aspects. So he's the one who started this in 1880. So he published several papers over a course of several years. He published all his findings, which have actually become very classical findings right now. Then the other person who also used the histology or histological tradition was Ramoni Cajal. So Ramoni Cajal, he said that these axons, the tip of these axons is actually a very specialized structure. So he came up with this idea again from looking at stained tissues, that this particular tip of the axon is called, he named it as axonal growth cone. And he said that this axonal growth cone is actually responsible for the growth of the axon and its movement to the target tissue. So if you imagine a big traffic jam, this axonal, the tip of the axon, which he called as axonal growth cone, that actually navigates through this traffic jam to reach the contact cell, the target cell, and establish the contact. So he said that this is probably how the axon is able to establish contact with the target cell. Next, so all of these studies, that Cahal's idea and William his observations, all of these were only by observing existing tissues. Say they could not actually perform any experiments as such. They were only making observations by looking at things under the microscope. So then what happened is, so this is called, like I said, histological tradition. So you just infer the mechanisms. But later, so there is someone called Ross Harrison. So Ross Harrison was the one who started experimental histological approach, meaning that he actually performed experiments using the tissues to come to conclusions. He did not just observe existing tissues, he also performed experiments with them. He was the one who started in vitro studies actually. So he was the one who pioneered what we call as in vitro studies. These days, in vitro studies have become very common, correct? But at that time, that concept was not there. So he is the one who started the experimental histological approach or the in vitro studies. So he addressed the so at that time there was other people who were saying that no, axons are actually just formed by the clumping of glial cells. The coming together of glial cells is what is actually axon. There isn't actually, axons are not things that grow from the neuronal cell, is what some people were saying. that they were claiming that neurons have thread-like connections with each other, and those that is actually what these other people have seen, is what other people were claiming. But Ross Harrison, he actually performed experiments and he showed that there is actually something called axon, and that axon is actually it is actually an elongation of the neuronal cell. And these axons are actually growing, and they actually have an axonal growth cone, and they try to establish connection with other neurons. So this is his main finding, that axons solely arise from nerve cells. He disproved the common belief that some people had, that axons are coming from the clumping together of glial cells. So he showed that that is not correct. He showed that axons come from nerve cells. So how did he do that? He did a very smart, elegant kind of experiment, and he took it. He was it was very difficult for him actually. It was not easy. Just a minute. Yeah. So it was not actually easy for him. So yeah. So Ross Harrison was someone who he studied in Johns Hopkins. Then he went to Berlin. So Berlin is in Europe, right? So it was like Europe at that time was like very full of, it was like a center for such very remarkable studies and experiments. So he was one of the few American biologists who first studied in America at Johns Hopkins. Then he went to Berlin. There also he studied, and then he came back to Johns Hopkins, and he tried to address this problem, that how is axon actually formed? Is it how the way some people are claiming? Is it actually from the nerve cell, or is it from glial cell? What is it? So what he did was, so he dissected small pieces of the neural tube. So neural tube is the embryonic, like it is the embryonic precursor of the nervous system. So that it is in the embryo, the neural tube is present, and the neural tube later gives rise to the CNS uh, of the of a person. So he took out neural tube. He dissected small pieces of neural tube from early frog embryos, and he took them and he kept in clotted frog lymph. So he wanted something to support the neural tube, because if he just kept them, they were not being sufficiently supported. So he kept them in a dish such that there were no other cells. It was only the cells of the neural tube that were there in that dish. And he added clotted frog lymph to as a solid, as a substrate in which the cells were supported, the neural tube was supported. And he used something called hanging drop method. So using that method, he tried to see whether these neural tube, whether actually axons are coming from the neural tube. And he did it in a time, like he checked it after different points of time. So this is what is actual, this figure is from his 1910 paper. So in that he has shown that in 24 hours, this is how it looks like. In after a few hours, you can see that there is this outgrowth, right? You can see that there is these small branches that are forming. Then he sees that it has extended further, and he sees that it has expanded and formed connections. It has moved, so it is able to move, like it can push ahead into the frog lymph, and it can move. So these outgrowing axons are actually forming growth cones, and that growth cones are extending into the lymph, that is the medium in which the neural tube was kept. And because there were no other cells, the glial cells were not there, right? So only the cells of the neural tube were there. So from that, he showed that these axons actually arise from the nerve cells, and that nerve cells are separate entities, and these nerve cells form connections with other nerve cells. So he showed that what is axon, what is the nature of the axon, and he showed that neural cells actually like to form connections with each other. So that is the very important experiment of that Ross Harrison performed, and he also established the early in vitro experiments that these days have become very common. Everyone these days uses in vitro studies, right? But at that time, Ross Harrison was the first person to pioneer the in vitro experiments. So that was what Ross Harrison did. Next, so now, the question of what axon is was addressed, but how does development happen? So development nowadays we know that there is a field that nowadays there is a field called developmental biology. We have a lot of understanding of development from blastula, gastrula, other stages of development, and so on. But at that point, people wanted to know whether there is any particular region in the embryo that is responsible, that can give commands to that can tell that okay, this has to become this kind of cell, this has to become this kind of tissue. So there is something that can help organize the embryo, so that can help that can act as an organizer. So people wanted to know whether there is in the embryo, whether there is something like that, whether there is a certain region of the embryo that can direct the formation of organs and tissues. So for people at the time were just speculating. They were not sure. They did not know how to test this. So at that time, there was also a lot of limitations. So right now, the methods that we have, the technology that we have is far more advanced. We have to understand that at that time, molecular level studies were very difficult. People were all doing things at the organ level. So molecular studies and cellular studies were like very difficult things to do. But still, that time people were so creative, they were so enthusiastic to understand how biology works. They did the kind of experiments they did and the creativity that they employed, we really should learn from that. So what they did was, so there was, like I said, there was limited experimental methods, uh, and everything was at the level of organs and tissues, and the most the at the early stages of for one single cell, two cell, four cell stage, it was still it was still easy to understand some things. But then in the later stages of development, it is like a multi like several cells are there. After the four cell stage, it became more difficult to understand what is happening. So what they did was till the four cell stage. So what I mean by four cell stage is that uh, we know that after fertilization, a single fertilized cell is there, that is the zygote, and then that divides to form two cells. Then those again divide to form the four cells. So that is the four cell stage, right? And then after that, there is again more division, and the blastula is formed. Then again, there is even more division, and other things happen, and the gastrula is formed, and so on. Just a minute. Yeah. So till the four cell stage, it was still easy that okay, there were four cells. So then what were till the four cell stage, how people tried to study was they tried to separate the cells by ligating the cells. Ligating means by joining or by tying this. Basically, they were making they were tying the cells by using human hair. Okay, so they used human hair is very thin, right? So they were trying to use threads or human hair or something to tie the cells so that they can separate the cells from each other. So they ligated cells by using human hair to tie the cells and see what is happening. So by ligating this, by basically, if they, let's imagine that there are four cells, They used human hair to to tie the cells so that they can separate the cells and see what is happening. But that was possible till the 4 cell stage. So they can use sea urchin eggs and they can tie the cells and see. But if the after the 4 cell stage, when there is like several cells, you cannot use these kind of methods, right? In that case, there were only two methods that were available to them. One method was ablation, means by destruction of some part of the embryo. And second was transplantation of one part of, like you can take one part, you can of the embryo, like slowly take it off from one side and then put it in on another side and see what is happening. So that was transplantation or grafting. So only these two methods were available to them to understand what is happening in the development. Now, even using these methods, they were very smart. So what did they do? There was a person called Hans Spemann at Wurzburg in Germany. So Hans Spemann was a very, at that point, he was a very big pioneer. He established a school in Freiburg. Okay, he established a school in Freiburg and he was studying. He developed various microsurgical techniques. He developed micropipettes and other things to understand what is happening in the embryo. So he came up with very, very cool techniques that even now people are actually using, advanced form of those surgical techniques that he had developed at that time. He was the one who developed micropipettes. So by, uh, by heating the pipette on a burner and then by using that to make very thin pipettes with which you can take the cells and the embryos and so on. So he came up with these microsurgical techniques and these micro level tools and techniques basically. So Hans P. Mann, what he did, so, like I said, he established a school in Freiburg, and some of his famous students will come to these people. So, Victor Hamburger was one of his students who would also contributed a lot. We'll slowly come to that towards the, like, as I go on. So, Hans Riemann, he had a PhD student. That PhD student was Hilde Mangold. So Hilde Mangold was working on newts. So newts are a type of an amphibian similar to they belong to the same class as lizards and salamanders and so on. So his student was Hilde Mangold, PhD student. So he told her that why don't you try to understand what happens when a certain part of the embryo is taken from one place and from one embryo and put in another region in another embryo. I'll explain that in detail. Just a minute. So, this one. Wait, I think I have a better image for that. Yes. So, he asked Mangold that you take an embryo of one type, so from one type, one species of that of newt. I said newt is also newt is an amphibian, right? So one embryo of one type of newt in which it is non-pigmented. Okay, so the cells are non-pigmented, and in another one, another embryo in which the cells are pigmented. So you can differentiate which is what. Because the one in one species the donor is non-pigmented and the other species it is pigmented. So that way you can distinguish the which cells you are looking at, pigmented versus non-pigmented. So he said, just a minute. So he asked her that why don't you take so in the early last stage, what happens is there is something called blastopore that is formed. Okay, a pore like region is formed, and how that happens is the cells, so the pore that is formed is called blastopore. Okay, and what happens is when the blastopore is formed, the cells migrate towards that blastopore, and then they enter inside they go to the interior of the embryo, and they from that the organs are formed. So basically, when the blastopore is formed, after the formation of the blastopore, the cells at the surface of the embryo, they move to the blastopore, they go to the interior of the blastopore, and they form the different organs of the body. Okay. So now what he asked her to do was, he asked her that you take cells from the blastopore, that is the from the dorsal lip of the blastopore to be exact. He said you take cells from one embryo which is non-pigmented, that is the donor embryo, and he asked her to graft or the transplant those cells to another to another embryo, that is pigmented. So this second embryo is going to have its normal blastopore also. Plus, Mangold is grafting the extra blastopore region to it. Just a minute. So Mangold is grafting this, is transplanting this dorsal lip of blastopore to the recipient embryo. So now there are basically two dorsal lips that are there, right? Two dorsal lips that are there, and because of that, there is a primary embryo that is being formed because of the original dorsal lip. Plus, this transplanted dorsal lip is leading to formation of a secondary embryo. And finally, what is happening is there are just a minute. And because of that, what is happening is two tadpoles which are joined together are being formed. So now from this, what did they understand was that this dorsal lip of the blastopore actually has some information that can act as an organizer, that can tell the embryo that this is how the uh, organs should be formed. So that information is present in that region. So it can act as an organizer. It can organize the fate of the embryo. So this was one important finding that they had. And another important finding was that these this transplanted tissue can have an effect on the neighboring existing tissues. So this transplanted tissue can affect the fate of the neighboring tissues. So that is called the organizer principle. So it became the Spearman-Mangold organizer principle. And they published this article it was a German article. They published it in 1924. But then unfortunately what happened is, so Mangold, as just after she submitted her thesis, uh, she died of a accidental fire, and because of that, it was only Hans Spearman who got the Nobel Prize, because Nobel Prize is not awarded after like it is not awarded after the person's death. So it was Spearman who received the Nobel Prize, although it was Mangold, his PhD student, who did the experiment and everything. But because she died, Spearman received the Nobel Prize for this organizer principle. which basically says that there is some information that is present that each for each organ system, there are certain regions that have the information that tell how that particular organ has to develop, or how that particular, so that for this has been shown for as other systems also. At the level of different systems, this has been shown that there are regions that can act as organizers. So that was the contribution of the work that was done by Hilde Mangold and Hans Spemann. So again, just I hope you are able to appreciate that all of these experiments were all done at the level of organs and tissues. These days we are talking about, single cell, single nucleus, those kind we are studying at that level. At that time, people did not have the methodology or the techniques to study at that level. But still, they were able to find you know, such important discoveries they were able to make. I mean, currently we have so many resources, we have so many developed techniques, but are we actually able to, make the same, extent of discoveries that those people did? Because at that time, although they were working in the level of organs and tissues, they were creative enough and they were smart enough and they worked hard enough to actually uncover these things, which at this point, I mean, at our current age, we are probably not, thinking to that level of creativity to understand what is happening, although we have even more advanced techniques at our disposal. So that is something we need to actually learn from these pioneering experiments. So basically, so far we have discussed about the axonal growth cone and the organizer principle. So till now, does anyone have any doubts? Is there anything that you want me to clarify or discuss? Or maybe something you didn't understand and you want me to explain it again? You can just unmute yourself and let me know. If not, then I will go on to discussing the next set of concepts. Okay, since no one is
Clear.
okay, very good. So let me just discuss some other important experiments, and then we can discuss the assignment questions. Okay, so yeah, so far we were discussing about the just a minute. So far we had discussed regarding the uh, organizer principle and the axonal growth cone. Now, this only explains that there are certain regions, right? But then are there certain molecules that are able to, dictate the development of the nervous system? What is happening at the molecular level? How do the cells of the nervous system know that what has to happen, whether they are supposed to grow, and how are they supposed to connect with each other, how are they supposed to grow, what governs all of that? That was the next question that these early investigators were grappling with. So they were dealing with how to find those things. So then at that time, what happened is, so that's what the question was: what is the relation between the developing neurons and the target cells? How do the neurons know that where to go, how to go there? All of these questions. So at that time, Frank Lilly was a pioneer in embryology in the US. So, like I said, there were but the Spearman and Mangold were there in Germany, that is Europe. In the US, Frank Lilly was one of the pioneers in embryology. So, Frank Lilly had a student. Margaret Sheroy. So Margaret Sheroy, he asked her that, just a minute. He asked her that why don't you check what happens if the limb bud is removed. So he said that you remove the limb bud of the. So he said that you remove the limb bud of developing chicks. So in the embryos of chicks, the limb bud is basically the precursor of the limbs of the chick, right? So he said remove the limb buds and see what happens. So she did this for both the embryos of chick and for salamander in 1909. And when she did that, she saw that she has removed the limb buds. She has not touched the nervous system, correct? But removal of the limb buds affected the cells of the nervous system. So both the sensory ganglion cells and the motor cells in the spinal cord, she saw that there is a reduction. So from this, what they understood was that there is probably some substance in the muscles. So the limb buds are giving rise to the limbs. So the by removing the limb buds, the muscles are going to get affected, right? So from that, they are inferred that there is something probably in the muscles, that some substance is there, and that substance is actually required for the survival or the growth of the neuronal cells. So that was something that Margaret Sheroy saw. Then, now we spoke about Ross Harrison, correct? Ross Harrison, who did the in vitro experiments and showed what is the nature of the axon. So Ross Harrison had a student called Sam Detweiler. So in 1920, several years after Margaret Sheroy's experiment, Sam Detweiler did a similar kind of experiment. So what did he do? He took salamander embryos, and in the salamander embryos, he grafted new limb buds. So Margaret Sheroy had removed the existing limb buds. But Sam Detweiler, he grafted new limb buds to the salamander embryos. And now, interestingly, he saw that if he grafts new limb, like an extra limb bud, that results in increase in the sensory cells. So basically, uh, if you get rid of the limb bud, that is reducing the neuronal cells. If you graft new limb bud, that is increasing the neuronal cells. So there is some influence on of the muscle or the limb buds on the nerve cells. So that is the experiments that were done by Sheroy from Frank Lilly's lab and Sam Detweiler from Ross Harrison's lab. Ross Harrison's lab. So these were two observations that were there, but people did not know that what is actually happening. So what is actually the influence? What are the substances that are responsible for this influence? So from this, what hypothesis was that there is some substance that is there in the tissue, muscle tissue, that is affecting the nerve cells. So the number of nerve cells is increasing or reducing if you do something to the muscle tissue. So this was there for several, like these observations were lying around for several years. But then, remember we had spoken about Victor Hamburger. So here we had discussed that Hans Spemann had a student, a brilliant student called Victor Hamburger, right? So in Hans Spemann's lab, from Hans Spemann's school, Victor Hamburger was a brilliant student. So he knew how to use these microsurgical techniques, how to do all of this. Like, to some extent, he knew all of this. In the newt cells, he had some experience. So now Victor Hamburger went to Frank Lilly's lab. So Frank Lilly was working with chick embryos, right? So Victor Hamburger went to Frank Lilly's lab, and Frank Lilly told him that, look, one of my students 20 years ago, she had done these experiments. She had seen that if you remove the limb bud, there is reduction in the nerve cells. If you and another person has shown that if you graft extra limb bud, there is increase in nerve cells. Do you want to do this? Do you want to follow up these experiments? So Victor Hamburger said, okay, let me try. So what he did was he followed up on these experiments, and he came up with the some certain hypothesis. So what did he come up with was, so he, like I said, he was a student of Spielman at Freiburg. He went to Chicago to Frank Lilly's lab. He wanted to learn neuroembryology, and Lilly told him to begin his studies to explore the uh, Margaret Sheroy, Sam Detweiler's experiments. Just a minute. So now Victor Hamburger, he also did further experiments with the chick embryos, and he saw that what Sheroy had seen was right. So if you remove the limb bud, then there is reduction in the number of both sensory neuronal cells and motor neuronal cells in the spinal cord. So if you're doing something to the muscle precursors, that is affecting the neurons in the spinal cord, right? And then he saw that he can actually quantify this. So if the number of muscle cells are reduced, there is an if x num if there's an x% reduction in the muscle cells, he can see a particular reduction in the neuronal cells. If you increase the number of muscle cells that are removed, there is even more increase in the number of neuronal cells. So there is a quantitative relationship. So if you remove more muscle cells, even more neuronal cells are affected. He saw this. And by seeing this, he came up with the following hypothesis. So Victor Hamburger said that the muscle, or any other target, whatever the sense organs are there, which are the targets of the neurons, these sense organs, including the muscles, they are probably generating something. They are probably making some substance. That substance is affecting the neuronal cells. So maybe there is one agent that is controlling the uh, spinal ganglion, and there is another agent that is controlling the motor neurons. Then he said that these agents or these substances are able to travel. So they are made by the sense organs or the muscle, and they are taken up by the nerves, and they are traveling into the nerves, and they are traveling all the way to the either to the motor columns or to the spinal ganglia. So they are made in the muscles or in the sense organs. They are taken up by the nerves, and they are going all the way to the region that is getting affected. And he said that this acts in a quantitative way. So um, if you reduce the number of muscles, that is basically if you take away more of the muscles, more of the nerves are getting affected, because the substance that is being produced by the muscles is getting reduced. So this is what he suggested. This was his hypothesis. Many years later, it was actually found that there is something called nerve growth factor. There is a protein called nerve growth factor, and this nerve growth factor actually acts like this. It is actually produced by several types of cells. It is actually taken into the neuronal cells. and it actually affects the both the growth of the neuronal cells as well as the direction of the growth of the neuronal cells. So he came up with this hypothesis years ago, but later it was actually shown that there is actually something that acts like this. So now when Hamburger was doing these experiments, he formulated this hypothesis. Okay. But he had to also like he wanted to find out what this substance was. Hypothesis was okay. But he wanted to find out what is the actual nature of this substance. Again, remember, this was all done at the level of organs and tissues. They did not have any molecular techniques like we have now. They could only remove things, they could only add things. They could only destroy a certain region of the tissue, they could only transplant some tissue. That was the only method that they could use at that time. Now, Victor Hamburger, he sent his paper to a person, a friend. That person was Giuseppe Levi. Okay, so he sent his paper to this his colleague Giuseppe Levi in Italy, and Giuseppe Levi sent it to his associate who was Rita Levi Montalcini. Now, Rita Levi Montalcini, so at the time there was war was happening. It was very difficult circumstances actually at that time in Italy. It was a very unimaginable time. So even during that time, Levi Montalcini actually managed to do certain experiments to find out what is happening. So she tried to do some experiments similar to what Victor Hamburger had done. And when she tried to do these experiments, she was not seeing the same kind of results that Hamburger had seen. So, whatever Hamburger had seen regarding what we have discussed so far, Rita's like conclusions are slightly different. Now, generally, what happens is nowadays, what happens? One lab publishes one thing, another lab publishes another thing. They both argue that I am right, you are wrong. Those kind of things, right? But Victor Hamburger, just see how these people are actually, the way people have been doing science, the way people have, the graciousness of these people, how they actually want to collaborate with each other to understand things rather than, fight with each other. So Victor Hamburger asked Rita to come over to his lab and work with him to find out what is the reason for this difference, what is happening, why are their results different. So Rita came over to work with Victor, and they found out that there is so we discussed that there is a when the limb bud is removed, there is a reduction in the number of neuronal cells, right, in the spinal cord. They found out that in general during development also there are cells which are naturally being killed, neuronal cells which are actually dying. So it is not that all the neuronal cells that arise during development, or it's not that all of them, develop into adult neurons and they are present and make connections with each other. But during the course of development, there is a natural death of some neurons that happens. So it is not just the cell death in the spinal cord close to the region of the limb that was removed, but it also naturally happens over the course of development in general in organisms in other unaffected regions. So what this shows, so this was the first observation that there is actually naturally occurring nerve cell death that happens during embryonic development. So there is always a constant pruning of neuronal cells depending upon the requirement. So that it's not like all the cells always survive till adulthood. There are cells that are pruned off, that are not, that are asked to die during development itself. This is the first time this was shown. So Hamburger and Levi Montalcini were the first to see this happen. Although they did not actually completely understand the gravity of that, but they actually observed that this happens. And then what happened is, so they had these hints that something is happening. They saw that there are these there are these outgrowths that are happening. There is this reduction in the cell number that is happening, and so on. But still, they didn't know what was the substance that was causing it. These are all just observations they had. Next, while they were searching for how to find out what kind of preparation to use to find out this substance, Victor Hamburger received a paper from his former student, who was Elmer Booker. So Elmer Booker, he had found something interesting. So what did Elmer Booker find was, so wait, let me just go here. Yeah. So what did Elmer Booker find was that he had put mouse sarcoma. Okay, sorry, here, huh? He had put mouse sarcoma. Sarcoma is basically cancerous cells, right? So he had put mouse sarcoma into embryos, and he was seeing whether that will affect the development of the embryo. Okay, so he had implanted tumors into an embryo, and he wanted to know whether such tumor will hamper the development of the embryo or whether it will help the development of the embryo. He had done this in chick embryos. So when he implanted the mouse sarcoma, to his amazement, Booker saw that it actually stimulated the growth of sensory nerves. So there was something that the tumor cells were secreting that was able to actually stimulate the growth of sensory nerves. Now, Booker did not know what it was. So, Hamburger asked Booker that, can I pursue this? Is it okay that if we pursue this, like whatever findings you had, can we pursue it? Can we find out what happens? So Booker said, all right. Booker said, okay, you can go ahead and pursue it. Initially, although he said it was all right, apparently later he regretted it. He thought that it would have been better if he had pursued it himself. But anyway, he consented to them using his approach. And then, so Victor Hamburger and Rita Levi Montalcini, they used Booker's approach. So they also checked whether if they placed a if they implanted a tumor next to a next to the dorsal root ganglia, does it actually stimulate the uh, outgrowth of the nerves? And they saw that actually it happens. So if the tumor is secreting something, some substance that is being secreted by the tumor, that is stimulating the growth of the nerves. So they said, okay, for now, let us call this hypothetical substance nerve growth stimulating factor. So next they wanted to find out, they wanted to purify this nerve growth stimulating factor. So they thought, okay, these tumor cells are able to secrete this nerve growth stimulating factor. Let us make extracts of the tumor cells. So they tried to. So Rita went to a person called, so a friend called H. Meyer. He was in Rio de Janeiro, Brazil. So Rita said, I will go to that person's lab and I will try to find out what is happening. So she went to Meyer's lab and she tried to do some tissue culture assays to find out uh, what is happening. So she tried to check the so she tried to establish a semi-quantitative way so that if there is if more tumor extract, more growth, less tumor extract, less growth, that kind of thing. So that was something that Rita was trying to do. On the other hand, there was Stanley Cohen. So Stanley Cohen, meanwhile, was a postdoc. He was a biochemist. He joined Victor Hamburger's lab. Okay, he joined Victor Hamburger's lab, and he thought, okay, let me try to do a biochemical way of understanding. So he took the tumor extract, and tried to do a chemical analysis to see that what it contains. But the tumor extract contained both nucleic acids and proteins. Now, how will you identify what is the what is the how which is the cause? So he said, okay, let me try to remove the nucleic acid. And how do you remove the nucleic acid? We will use phosphodiesterase. Just a minute. Yeah. So he said, okay, let me use a phosphodiesterase enzyme, which will basically degrade or cleave the nucleic acids. And only the protein will be there, and I can purify the protein. But when he exposed the tumor extract, so he said, where will I get the phosphodiesterase? So people knew that snake venom had phosphodiesterase. So he said, okay, let me expose the tumor extract to snake venom. So that will result in the cleavage of the nucleic acids, only the proteins will be left. And I can purify the protein. But the problem was that when he used the snake venom to this tumor extract, the venom itself is able to stimulate the cells. So when the venom by itself, the snake venom by itself contained some growth factor that can stimulate the neuronal cells. So then they thought, okay, instead of looking at the tumor extract, let us try to purify the growth factor from the venom instead. So snake venom has growth factor that is also able to stimulate the growth of the nerve cells. Let us try to purify the protein from the snake venom. Now, snake venom is secreted from the salivary gland. So they thought, okay, then we will investigate the salivary gland. But they investigated the salivary gland not of the snake but of the mouse. So they took the salivary gland of the mouse. They tried to purify they took lots of salivary glands and they saw that it has a lot of growth factor. And from that, they purified the nerve growth factor. But they were able to purify the nerve growth factor, but they did not know the molecular nature because at that time that had not come up yet. So much later, uh, Angelity and Bradshaw, these two people, this was much later, these two people used the several submaxillary glands, the salivary glands of mice as a source, and they sequenced the nerve growth factor protein. So this is the whole. So this took several years of research for them to identify that there was a hypothetical. Initially, there was a hypothetical substance that can affect the growth of nerve cells that is secreted by muscle cells and sense organs, and that can affect the growth of the nerve cells. And then the actual experiments they did to purify the nerve growth factor, then the sequencing. This was a work of several years. And nerve growth factor was the first growth factor to be identified. And it is also the most, till date also, it is the most widely studied, best studied growth factor. Now we know that there are several other growth factors also that have people have studied. But nerve growth factor was the first discovered one. And this is how it happened over several years and several, continents. It happened. And Hamburger and Levi Montalcini, they got Nobel Prize for this work for the discovery of the nerve growth factor. And now let me just introduce these two terms. One is neurotropic action, and the other is neurotrophic action. So neurotropic action is something that affects the direction of the growth of the neuron. So the direction in which the axon is going to move, the axonal growth cone is going to form the contacts. That is the that is affected by a neurotropic action. Neurotrophic action is one that is necessary for the survival of the neuron itself. Like we discussed, some neurons are dying during development, some neurons are able to survive. So the neurotrophic action is the one that dictates which neurons will die and which neurons will survive. So these are two different things that happen. One is the direction of growth of the neurons, and the other is phenomena that affect the survival of the neuron itself. Interestingly, NGF or nerve growth factor that we discussed actually has both of these. Not just NGF, there are several other growth factors also that can actually do both of these things. It is not true necessarily true for all factors, but many of them act like this. That they can have both neurotropic action as well as neurotrophic action. So they can affect the survival of the neuron itself, and they can also affect the direction in which the axon is actually extending, that the direction in which the axon is growing to reach its target cells. So this is all about this I thought was very important because nerve growth factor is the discovery of nerve growth factor is very interesting and very, it happened over several years and because of several people. So I wanted you to highlight how the nerve growth factor actually works. I mean, how it was actually discovered. So this was how the nerve growth factor was discovered. So far, is there any questions that anyone has that you want me to address? Or any doubts that you have? Or maybe anything you want me to recap or something you didn't understand? Okay, so since no one, uh, I believe no one is having any doubts, let me move on to the uh, next part. I think I will just briefly talk about some of the other things that were discussed in this week's course, and then we'll discuss the question answer segment because there's not much time. I'll just quickly go through some of the important things that were. Just a minute. Hmm. Yeah. Uh, Let me also talk to you about the about the theories and the experiments that were done. So we had spoken about the organizer principle, right? So we know that there are certain cells or regions that are able to organize the formation of the organ or the tissue itself, right? But what actually how does what is the way in which these organizer regions are organized? How do you know that? How do the cells know that how to make the contacts? So there is a there is one cell that wants to make a contact with the target cell. But what are the factors that determine the specific connections between those cells? Are there any factors? Are there any substances that guide the guide one of the cells to establish contact with the target cell, or are there no actual factors? So at the time, people didn't know that, and they came up with different theories. So John Langley came up with one of the chemiotaxis theory. So he said he basically cut preganglionic fibers of the spinal cord, of the superior cervical ganglion, and he these generally undergo regeneration. Okay, so he first cut them, and then they underwent regeneration. Then he checked whether the re-innervation was the same as the original innervation, or was it different. And he saw that the re-innervation was extremely precise. So the way it was before the fibers were cut, it was the same way even after the regeneration of the uh, ganglionic fibers. So he said that there are probably precise chemical interactions. There are very ordered arrangements, and these probably arise because of precise chemical interactions. He said that there are probably some chemicals that are maybe acting as cues, and that are helping the cells to know that this is how I should form my the connection with my target cell. So that was John Langley's experiments and his chemiotaxis theories. So these are one of the earliest theories. This was in 1890s. So nerve growth factor had not been discovered at that time, remember? Because nerve growth factor came much later. It came in the 1950s. Whereas what we are talking, John Langley's experiments happened in the 1890s. So at that time, actually, William Hiss had just made his observations about the fact that there is axon and there are dendrites and all of this. So at the same time, John Langley was putting forward his chemiotaxis theory. At that time, nerve growth factor had not even been discovered. There was no hypothesis about a nerve growth factor. So Langley made these speculations, but then that remained that way for several years. Then Paul Weiss. Paul Weiss was from Vienna. So Paul Weiss, he was from Vienna, but he also worked at Chicago and at Rockefeller University. So Paul Weiss in 1920, he said that there is no it is not it is so he said that this is because of resonance. So the way a violin, if you touch a string of a violin, the way it undergoes resonance, that is how the muscle contract. So okay, let me rephrase this. So what he did was he said that if you graft muscle tissue, okay, from one organism to the other, the grafted tissue, the grafted muscle is also able to perform muscle contraction, similar to the host tissue. So the host tissue is performing muscle contraction. Similarly, the grafted tissue that has come from another organism and it has been transplanted to that particular place, it is also able to perform muscle contraction. He said that the innervation of the nerves into this muscle is random. It does not follow any specific pattern. The innervation of the host nerves into this grafted transplanted tissue is random. So he said this is random, the muscle, the transplanted or the grafted tissue is also able to contract because these muscle tissues probably have some, um, they respond to specific impulses, and that is the reason they are able to contract. Okay, he said that this has nothing to do with any chemicals that are released. This is probably because the grafted tissue is encoding some memory of some impulses. So it knows that if there is specific this kind of pattern of activity from the nerve, it has to contract. So that is how it is able to contract. It is all based on the impulses. And he gave an analogy to the way the strings of a violin work. So that is why this theory is called resonance theory. He said that muscle contraction depends completely on temporal activity, patterns of activity, rather than which space or which play. It's not a spatial thing, it's only a temporal thing. It is only based on the impulses from the nerves. So, over time there has been, like, questioning of the theory, resonance theory, whether it is actually true or not. There were a lot of discrepancies that were observed, because now we know, right, that muscles contract. So depending upon the, so we know that for one particular muscle, like one particular type of muscle to contract, that motor neuron has to, like, there has to be activity from the motor neuron to that muscle telling it to contract. So that is how muscle contraction happens, right? It's not like all the nerves throughout the body are active all the time. It is not like that. For that particular muscle to contract, the nerves that are innervating that muscle have to send the correct information to the muscle. That is how it happens. So that is why later Paul Weiss's resonance theory came into a lot of question. Even now, people are trying to, like, people are thinking about whether in some context it may be true, in some context it may not be correct. So that is the resonance theory of Paul Weiss. Next comes the contact guidance theory. So contact guidance theory was again from Paul Weiss's experiment. So he said that there are no factors or cues. The axons are just bundled together. And because of the bundling of the axons, they know that where they have to go. The axons are all bundled together. There is a physical contact between them. And because of the physical contact between them, they know that what direction they have to follow. There are no chemical signals that are involved, is what uh, Paul Weiss's contact guidance theory said. Finally, was Roger Sperry's chemoaffinity hypothesis. Now, this is very important because Roger Sperry uh, was Paul Weiss's PhD student. Okay, so he was he did his PhD with Paul Weiss, and then he did his postdoc with Carl Lashley. Okay, and then so both Paul Weiss and Carl Lashley were not. So Paul Weiss, like I said that there is no specific chemical signals. Carl Lashley also did not believe that there is any specific functional aspect to the different parts of the brain. But Roger Sperry, who had worked with these people, came up with a completely opposing theory to his PhD and postdoc advisors. He said, so he did a very, very, very elegant experiment. He used the connection between the um, retina of the eye and the optic nerve, right? So the retinotactile pathway it is called. He made a cut over here, or a transection. He made a cut of this optic nerve. He rotated the eye to 180 degree. Okay. He cut this nerve, he rotated this eye 180-degree, and then he saw what happens. So this optic nerve is can regenerate in these animals, in newts, goldfish, and frog. The regeneration is possible. So the optic nerve regenerates and it goes and attaches to the retina exactly the way it generally does normally. But Roger Sperry had rotated the eye by 180-degree, right? So now the eye is rotated. Because of that, and but the optic nerve is forming the exact same connection as it generally does. So after regeneration, the exact same connection, exact same to same connection was formed. Now, because of that, the animal, like the animal thinks that the animal is viewing the world in an down manner, and the animal is behaving also like that. So from this, what Roger Sperry showed, or he concluded, was that the both the neuronal cells and their target cells, they have specific biochemical identification tags. Just a minute. So they have specific biochemical identification tags, and that is responsible for the specific connections that exist. So he said that he called this the chemi-affinity hypothesis. So basically, if you see the chemi-affinity hypothesis, it is very similar to the chemiotaxis hypothesis that John Langley had proposed. So basically, this is kind of, after several years, again, we can say it is in the golden decade, right? In 1940s, 1950s time, the chemi-affinity hypothesis came into existence. And basically, using this hypothesis, Roger Sperry, his hypothesis was complete opposition to both his PhD and postdoc advisors. But even now, several of the like studies that we have and several of the hypotheses we have are some variants of this chemi-affinity hypothesis. So what Roger Sperry proposed actually in some way is still guiding our current studies. So later, like lot of refinements to the chemi-affinity hypothesis have happened over the years, and even now people are trying to understand what is the like how this kind of how what is the axon guidance that is happening, how axons know where is their target cell and how do they go and what are these guidance molecules, axon guidance molecules that are helping this to happen. So even studies in Drosophila have shown that not just in Drosophila, even in C. elegans, for example, they have experiments have been done and shown that there are guidance molecules that exist which are able to like help the axon reach its target cell and form a specific connection. And then there are olfactory receptors have been shown to be specific molecules that can guide axons to their specific glomerular targets. So these are some of the things that were done. So this is how the connections themselves were organized. So does anyone have any specific doubts? Is there anything that I you would like me to discuss over these? Okay. Maybe I can first now discuss some of the question answers, previous year question answers, and then come back to the concepts depending on the time we have because we need to discuss this also. So, would any of you like to answer? Like why? Uh, so, one second. Yeah. So I think So we started with this, right? We started with the fact that William His was the one who started the study, who is the founder of neuroembryology, and he was the one who did the observations of different embryos, so even specifically human embryos of different ages. He observed them under the microscope, he stained the fetal tissues, and he observed the neuronal development. That is why the correct answer to this question is Wilhelm His. Does anyone have any question? Someone's Prem, do you have any question? Prem, could you please mute yourself? In case you don't have any questions, could you please? Okay, Prem left. Yeah. Next, what did Ramoni Kahal call the tip of the axon? So, like we just discussed, he said it is called axonal growth cone. And we now know that the axonal growth cone is actually a specialized structure. It is rich in the actin cytoskeleton and it helps the axon to reach to its target cell. So, the correct answer to this question is axonal growth cone. Next, select all the incorrect statements regarding Harrison's experiment on axon formation. So, we know that Harrison was the one who investigated axonal growth in vitro tissue culture. So, like I said, he was the pioneer. So, of course, this is correct, right? Next, the experiment strengthened the belief in the coalescence of the glial cells. But Harrison's experiment disproved that. Harrison showed that it is axon is actually emerging from the nerve cells, correct? So this is the wrong statement. The experiment involved dissecting small pieces of neural tube from frog embryos. That is absolutely correct. So Harrison dissected the neural tube, which is the precursor to the CNS. He took those from the frog embryos and he kept them in a dish which contained clotted frog lymph. So then this is correct. Next, he validated the axonal origin from nerve cells. Yes. So when he did the experiment, there were only nerve cells in that dish. There were no other types of cells. And that is how Harrison showed that the axons originate from nerve cells and they are not from the glial cells. So that's why the incorrect statement is the second one. Is that clear?
Yes, ma'am.
Okay. Next. Mangold's experiments primarily focused on blastopore transplantation at the gastrula stage. So this is wrong because she did all her experiments in the sorry, she did it. One second, let me go there. So Mangold's experiments were all done in the using the blastopore, right? So that's why that particular statement is false. Next, select all the correct statements with regard to Mangold's transplantation experiments. Yeah. So select all the correct statements with regard to Mangold's transplantation experiment. So we just discussed that the first statement is wrong, right? Because the blastopore transplantation did not take place in the gastrula stage. It took place in the
Blastula stage.
Yes, very good. So next is uh, the formation of pore and migration of cells to begin organ formation. So Mangold's experiments, the blastopore which was forming, that had to how will this blastopore form? How will the so from the blastula, the blastopore forms. From the blastopore, sorry, when the blastopore is formed, the cells on the surface of the blastula migrate to the blastopore. Then when they move to the blastopore, they migrate into the interior of the embryo and then they form the organs. So basically, this is the mesoderm. So the mesoderm these are cells of mesoderm cells that belong to the mesoderm. They first move towards the blastopore, then they enter the interior of the embryo, and later the mesoderm cells form the internal organs. So that is why the correct answer is formation of pore, migration of cells to the pore and then to the inside of the embryo, and then followed by organ formation. And the third and the fourth statements are obviously wrong because the blastopore does not disappear. The blastopore is required for initiating the migration of the cells to the blastopore. And finally, because of the migration of cells to the interior, the organ development happens. And without the migration, of course, nothing else can happen. So the fourth statement is also incorrect. Next, what does nerve growth factor stand for? I'm quite sure that by this time everyone here knows that nerve growth factor is the correct answer. Next, match the following. We just discussed this. So chemotaxis theory was given by John Langley. So he is the one who performed the first set of experiments to understand how are the connections being formed. So he did this in the 1890. So he showed that he came up with the chemotaxis theory, and he proposed that there are some chemical interactions that exist between uh, between the nerve cells and their target cells. Next is the chemo affinity hypothesis. We have also again discussed that it was Roger Sperry who proposed this hypothesis in the 1940s. So he did the experiments with the optic nerve and the retina, and he showed that he came up with the hypothesis that there are some biochemical identification tags that are probably contributing to the formation of the connections. And the resonance theory was proposed by Paul Weiss, and in which he said that there is probably just temporal impulses that are governing the muscle contraction. And they might not, according to him, there isn't any chemical interactions as such. So these are the three different theories that were proposed by these people. So the correct option is 1, C, that is the chemotaxis theory, John Langley. 2, chemoaffinity hypothesis, Roger Sperry. And 3, resonance theory, Paul Weiss. Is that clear?
Yes, miss.
Okay. Yeah. Next. What did scientists do in their early experiments to understand axonal transport? So at that time, since they did not have any, I mean, nowadays we have experiments. I mean, we nowadays we have live cell imaging, we have fluorescent, we have antibodies that can label specific neurons. We have tags that can label specific vesicles. We have dyes that can label mitochondria. We have very nice genetic techniques that are in use. We have laser. We have so many different things, right, these days. But at that time, scientists did not have these access to such techniques. So they did it. The simplest way by which they tried to understand how axonal transport works is they tied or ligated neurons. So when they, if you make, if so, Paul Weiss, like I said, worked on axonal bundles, correct? So what Paul Weiss did was he basically tied the neurons. Sorry, tied the axon bundles. Just a minute, let me go to that slide. Yes. So Paul Weiss, with along with his co, both of them, they did a simple ligature experiment. So basically, by tying axonal bundles, they saw that there is a bulge on the um, on one side of the ligature, and they from that they inferred that there is a flow of materials from the cell body or the soma to the periphery or the synapse. So from this experiment, I mean, so this is the kind of experiments people initially tried to do. Later, you started using radioactive tracers, and nowadays we are able to do live cell imaging to actually see the movement of vesicles and so on. But initial experiments were as simple as just tying the axonal bundles. And now, I mean, now our experiments have advanced, and we know that there is anterograde transport, there is retrograde transport in the axons, there is transport of organelles like mitochondria, there is transport of vesicles, there is transport of mRNA. There's so many different things that are being transported across the axons, and that happens through the microtubules that act kind of like railway tracks. Next, in the 1860s, Schmeideberg conducted a study. Sorry, just a minute. In the 1860s, Schmeideberg conducted studies on a particular substance that mimicked the inhibitory action of vagal stimulation on the heart. So the correct answer for that is muscarine. So muscarine is a substance that can, muscarine is a substance that, so the inhibitory action of vagal stimulation on the heart is because of acetylcholine, and muscarine is a substance that mimics the action of acetylcholine. So the answer for this question is muscarine. Next, what did Paul Ehrlich explain using the lock and key analogy? So this is something that we had. Let me just briefly go through that. The lock and key hypothesis. I'm quite sure many of you would know, but let me still. Yeah, so this is the experiment that was done by for the previous question that we discussed. So he discovered he dis he discovered that there is an alkaloid substance that is present in Amanita muscaria, in this particular type of fungus, that can inhibit the that can mimic the inhibitory action of vagal stimulation on the heart. So at that time, we did not, I mean, there was not, pharmacology was not developed to that extent that it is right now. So at that point, these kind of discoveries actually what made us better understand that there are such molecules that can act on the nervous system that have these certain effects, and they are able to mimic the action of what is happening in vivo, and we can maybe use some of these substances as drugs, as therapeutics. They can help us, or maybe they can be poisonous to the nervous system. So these kind of things were slowly coming up. People were slowly trying to understand that what kind of molecules can affect synaptic transmission, what kind of molecules can cause like increase in synaptic transmission, decrease in synaptic transmission, inhibition, excitation, neuromodulation. So all of these things people are slowly trying to discover. So one of the things that was shown was that there is an alkaloid substance that is present in this species that can actually mimic the inhibitory action of vagal stimulation on the heart. So the correct answer to that question was that therefore muscarine. Now coming to the this question about what Paul Ehrlich did. So Paul Ehrlich, before Paul Ehrlich, there was Emil Fischer. So Emil Fischer had proposed just a minute. So Emil Fischer had proposed a lock and key hypothesis. Okay, so Emil Fischer had first proposed that the enzymes might actually act as a lock, and the correct substrate when it interacts with the enzyme is acts as a key, and that is how enzyme substrate interaction happens. Now, Paul Ehrlich, whose contributions were mostly in the field of immunology, he said that this is probably true even in the immune system. He said that there are probably proteins that act as a molecular nucleus. So proteins that have a molecular nucleus, and there are certain regions that are like similar to a lock and key model. There are certain regions that have specific interactions. So there is a molecular nucleus, and then there are regions to which specific molecules can bind, including particular antitoxins and so on. And that is what leads to the molecular activation of these proteins. So, and he specifically proposed this in the immune system. So that is why the answer to this question is that Paul Ehrlich explained the immune system specific interactions using the lock and key analogy. Next is, what was the term introduced by John Langley to describe the nerves associated with the cranial and sacral parts of the autonomic nervous system? So the correct answer for that is parasympathetic. So he said that these nerves that are associated with the cranial and sacral parts, he said these should be called as the parasympathetic nervous system. Next, Dixon and Dale observed two distinct actions in their identification of acetylcholine: a rapid effect mimicked by muscarine and a slow effect mimicked by nicotine. Is this statement true or false? So, does any one of you know why this statement is false? Can any one of you explain to us all? I will, of course, be correcting you in case you make a mistake, but would any one of you volunteer to explain why the statement is false? It is not, why is it not true? No? Okay, let me explain. So, Walter Dixon and Henry Dale, when they identified acetylcholine, the way they identified it was that they saw that acetylcholine has a very rapid effect in some cases, and this rapid effect was mimicked by nicotine, and its slower effect was mimicked by muscarine. But in this statement, it is said that they observed two distinct actions, a rapid effect by muscarine and a slow effect by nicotine. So that is completely the opposite. The rapid effect of acetylcholine that they observed was in fact mimicked by nicotine, and the slow effect was mimicked by muscarine. So the that is the reason why this question is right, this statement is false. So remember, acetylcholine has a rapid effect, and that rapid effect is mimicked by nicotine, and the slow effect of acetylcholine is mimicked by muscarine. So that is the observation that Walter Dixon and Dale observed. Next, the neurotransmitter identified in mammalian brain extracts in the 1950s, later confirmed as the major inhibitory neurotransmitter, is. So the correct answer to this is GABA. Okay, so GABA is an inhibitory neurotransmitter. GABA was initially identified, like it is said in the statement, it was initially identified in mammalian brain extracts in 1950s. But people were not able to understand what is the function of GABA. Let me just go to those slides. Just a minute. Yes, here the timeline for the timeline about GABA's discovery. So, GABA was first identified in mammalian brain extracts. It was identified in the 1950s itself, but the problem is that, not the problem. So, after it was identified, people were not sure about what GABA does. Some people were saying, so some people said that it is probably an inhibitory neurotransmitter. But many people were skeptical. They were not convinced. Some people said that GABA, they said that there is very less evidence that GABA is an inhibitory neurotransmitter. They were not ready to believe that GABA actually is an inhibitory neurotransmitter. So, although it was discovered in mammalian brain extracts in 1950, it took around 30 years, more than 30 years, for people to actually accept that GABA is a inhibitory neurotransmitter. So, the correct answer for this question is that GABA is the neurotransmitter that was identified in mammalian brain extracts in 1950s, and then it was later, much later, it was confirmed in the 1970s and 1980s that it is a actually an inhibitory neurotransmitter. And again, we can see that then it is the 1950s, which is the golden decade in which the identification of GABA also happened. So time and again, we have been like the results. Our discussion is reinforcing the fact that the 1950s was the golden decade of for neuroscience. Next, select all the incorrect statements with regard to neurotransmitters. Okay, first, connection between serotonin and the hallucinogen LSD was observed in 1950s. This is a true statement because in the 1950s, it was actually discovered that the hallucinogen LSD actually, okay, let me just go to that slide. Yeah. So it was discovered that the hallucinogen LSD actually antagonizes the effect of serotonin. That means that whatever is the effect of serotonin, the effect of LSD is the opposite of that. So, and why is that happening? Because the administration of LSD is decreasing the content of serotonin in the brain. So, the connection between serotonin and LSD was discovered in the 1950s. Therefore, the first statement is correct that there was a connection that was observed between LSD and serotonin in 1950s. First time it was observed in 1950s. Next, first evidence of acetylcholine in the Renshaw pathway acting as an excitatory neurotransmitter. Yes, acetylcholine acts in the Renshaw pathway, and acetylcholine also acts as an excitatory neurotransmitter in that pathway. So, what is the Renshaw pathway? Let us just go and quickly discuss about that. Sorry, just
Madam, one doubt. Can you
yes.
please explain LSD antagonist once more?
1 minute. Let me just go there. Yes. So, what they saw was that so LSD is a hallucinogen. Even now, it is people use it as a hallucinogen. It's a hallucinatory drug. So initially, people saw that there is a connection between serotonin and LSD. Later, they came to understand that LSD so whatever is the effect of LSD, that is the opposite of that of serotonin. So serotonin stimulates the smooth muscle. Okay, serotonin acts on the smooth muscle and it stimulates it. But LSD prevents the stimulation of the smooth muscle. Okay, so LSD acts in the opposite manner to that of serotonin. Okay, that is the observation they made, but they did not know what is happening. Then they saw later in 1957 that when LSD is administered, that is reducing the content of serotonin in the brain. So that is how LSD is actually acting. LSD is reducing the brain serotonin content, and because of that, the stimulating action of serotonin is lost. So LSD and serotonin are antagonists, act as antagonists to each other. So that is how LSD is also able to cause the hallucinations. So I will maybe in my next class I will uh, come up with a slide to explain this in greater detail, like what is actually how it was done and what is actually done. So, or maybe I can post it in the discussion forum. So right now, I do not have a PowerPoint slide to explain it in better detail. But may I know who asked the question?
Ashwini.
Okay, Ashwini. I will, in the next class, I will come up with a PowerPoint slide to explain the exact way the experiment was done so that you can understand this LSD and serotonin connection in greater detail. Okay. Sorry, I don't have a actual PowerPoint slide to explain it, but in the next class, I will come up with a slide to explain how the interaction between LSD and serotonin.
Okay, ma'am. Thank you.
Okay. Yeah, so I was talking about acetylcholine and Renshaw pathway, right? So let me just go over there. I think I skipped. Oh, it's in the previous. Yeah. So acetylcholine. is so there are certain cell interneurons called Renshaw interneurons. Okay, so acetylcholine is secreted by the alpha motor neurons, and acetylcholine when it is released by the alpha motor neurons, it is an excitatory neurotransmitter that stimulates the muscles. Okay, so it is released by the alpha motor neurons. It acts on the acetylcholine receptors that are present on the muscles, but it also synapses with the Renshaw interneurons. So now the Renshaw interneurons, what they do is they inhibit the they inhibit the motor neurons. So they have a feedback mechanism in which they inhibit the alpha motor neurons. So, from like Dale's law, we know that acetylcholine is an excitatory neurotransmitter that if it is acting excitatory on the muscles, if it's released from the motor neurons, it is an excitatory neurotransmitter. It is stimulating the muscles. That means it is also stimulating the Renshaw interneurons, because if it is stimulating in one synapse of the at one synapse formed by the axon, it will act as an excitatory neurotransmitter in all the other synapses also. It is going to act the same way at all the synapses. So all the synapses of that particular motor neuron are going to, in which acetylcholine is released, is going to cause excitation. So, acetylcholine also forms synapses with Renshaw interneurons, and it acts as an excitatory neurotransmitter at those synapses. And the Renshaw interneurons get activated and they give a feedback inhibition on the alpha motor neurons, which are actually secreting the acetylcholine in the first place. So, in the Renshaw pathway, acetylcholine is acting. So that is why that particular statement that there is acetylcholine acts in the Renshaw pathway as an excitatory transmitter is correct. Next, initial skepticism and a lack of evidence for GABA causing inhibitory postsynaptic potential. So we discussed this, right? That people were not very people were very cautious. They were not readily accepting that GABA is an inhibitory neurotransmitter. So there was a lot of initial skepticism. That is true. And there were also people, the reason they were not accepting was that they thought that there is a lack of evidence that for GABA to be an inhibitory neurotransmitter. So this statement is true because GABA was after several years after its discovery, GABA was actually accepted as an inhibitory neurotransmitter. It was after several decades it was accepted. So the third statement is also true. So then what is the incorrect statement? The incorrect statement is that glutamate was acknowledged as the primary inhibitory neurotransmitter in the brains in the 1980s. Glutamate is not an inhibitory neurotransmitter. Glutamate is an excitatory neurotransmitter. So that is the reason this statement is false. Glutamate is a primary excitatory neurotransmitter in the brain. GABA is the primary inhibitory neurotransmitter in the brain. So that is why the fourth option is the correct answer for this question. Next, what did Claude Bernard discover about the arrow poison curare? So curare, so Claude Bernard discovered this from so in South America. Let me just go to that. So in South America, there is this arrow tip poison called curare. Okay, so now Bernard, it was known as a poison, but people were not able to understand why it is a poison. So Claude Bernard did work with this Curer, and he showed that when this Curer is applied to nerve and muscles, it causes paralysis. Okay, and because I mean, it was shown that it is a poison. It is causing paralysis. So now, keeping this in mind, let us go back to the question. What did Claude Bernard discover about the arrow poison curing? He discovered that it causes paralysis when applied to muscles and to nerves. It is not completely harmless. It is actually a poison, but the way it was acting was not known previously. But Claude Bernard showed that it causes paralysis of the nerves and muscles. Next, who first talked about the lock and key idea? So I think you all already know the answer to this question. The answer is Emil Fischer. So Emil Fischer was the one who came up with the hypothesis that enzymes have a lock and key in enzymes, and their substrate work via a lock and key model. He was specifically talking about the sugars, but that is actually the this actually applies for all the enzymes that are there. He was talking only about the sugar converting enzymes. But now we know that now there is something called a modified lock and key model, in which like later people realize that this is not just for sugar converting enzymes, but also for other enzymes also. So almost all enzymes have an active site. They have a pocket to which the substrate will go and interact. And only like depending upon the shape of the catalytic, the active site, and the shape of the substrate, the specific interactions happen. So the lock and key concept was proposed by Emil Fischer. Next, what was a key methodological advancement in neuroscience during the 1950s? First statement, studying brain activity at the organ level. That was not a key methodological advancement in the 1950s, because even before that, that is how people were studying. We know, right, that Wilhelm His was looking at the embryos. Then after that, other people were looking by transplanting the grafts, and then in the 1910s and 1920s and so on. That is how people were actually studying, like doing experiments in neuroscience. They were actually doing it at the organ level. So that is not a key methodological advancement in the 1950s that happened. Then creating textbooks about brain biochemistry. No, at that time, it was not like there were no proper books which spoke about brain biochemistry. In fact, the information at that time was very limited. People had just started understanding how brain biochemistry works. So textbooks were actually created like it took a lot of time for people to actually understand and come up with like bring biochemistry into the picture of with related to the brain. So no, creating textbooks about biochemistry was not a methodological advancement in the 1950s. Identifying only two neurotransmitters. No, people had by that time, they had in the 1950s, they identified many different neurotransmitters. So it was, let me just go there. Yeah. So although there was the information on neurotransmitters was limited, still by that time they had identified some of the neurotransmitters. They had identified acetylcholine, norepinephrine, glutamate. So they had identified some of them. It was not just two. So that is why that answer is, I mean, that statement is also incorrect. And what is the correct statement? The correct statement is that the key methodological advancement in neuroscience in 1950s was the development of the glass micropipette as an intracellular electrode. So people were starting to do electrophysiological experiments. So electrophysiology means to understand what is the electrical nature of the neuronal cells. So we know now that in the neurons, electrical activity happens. There is action potential, there is propagation of the action potential along the axons. There is at the synapse, there is electrical transmission of in chemical transmission of the information, and that again causes electrical information in the postsynapse to get initiated, and so on. So, for the study of electrophysiology had started in the 1950s, and that how was that being done? That was being done by using glass micropipette. So, people were using intracellular electrodes. So, you need a stimulating electrode and a recording electrode in order to uh, find out what is the electrical information that is being passed in the neuronal cells. And for that, glass micropipette was developed in the 1950s. And just a minute. So this was the intracellular electrode was developed in the 1950s, and it was a very, very important, it allowed us to basically start the electrophysiological experiments that even now there are glass micropipettes that are used as electrodes, and people try to understand even what happens in the case of disease. Is the like interest is the record other recordings different, what happens when certain drugs are given, is the other electrical signals hampered. So these kind of electrophysiological experiments were possible because of the development of the glass micropipette as an intracellular electrode in the 1950s. Next, we are almost near the end, so only two more questions to go. Okay, who discovered important evidence about dopamine in the brain? So this was done by Arvid Carlson. He got the Nobel Prize for that. He was the one who put forth important evidence that regarding dopamine, and he was a pharmacologist, and he contributed greatly to the discovery of dopamine, and he was the one who first did that. After that, Ole also came up with certain important evidence, but the first one to do it was Arvid Carlson. Next, and the last question for the day. So, what are pheromones used for? No, sorry, one more slide is there. Okay. What are pheromones used for? So, pheromones are chemicals that actually help in animals of the same species to communicate with each other. The pheromones are released even during mating and courtship, and basically they act as signals that help one animal of a species to communicate with another animal of the same species. So this was studied using, so it was first studied in insects, and it was discovered that there are these molecules that are able to transfer information, or they are help in the communication of animals of a particular species. So the correct answer is therefore to help animals of the same species communicate. The last question, I think we just discussed this. Where did scientists first measure electrical activity to study pheromones? So they used the insect antenna. So they developed an instrument called electroantenogram. Let me just go to the slides and show you where you can find the answer. Just a So pheromones are actually very interesting because they are present even in humans, and they affect a lot. I mean, sorry, they affect a lot of things. They even affect mate choice preferences. So they make us they influence which what like the mate of an organism with which it will mate, and it will uh, cause reproduction. Just a minute. So Electroantenogram was the instrument that was used to record insect antenna responses. So the to pheromones. So that is why the correct answer to that question is where did scientists first measure electrical activity to study pheromones? They used the insect antenna for that. Even now, animals, the fruit fly, Drosophila melanogaster, people carry out extensive studies to understand the effect of pheromones on mate choice preference, what is the effect during courtship, what have like how does it, what are the consequences of altered pheromones and so on. So with that, I come to an end of the class of the live interaction today. We have discussed several concepts that I thought were important for you to know, and I've discussed some of the previous year assignment questions. If anyone has any specific doubt regarding any of this, any of the past assignment questions or any of the concepts, feel free to ask right now, or you can post your question in the discussion forum, and I would be happy to answer your questions. And if you want me to discuss anything that you feel that was not covered in this session, I can discuss that in the next class. So feel free to let me know what are the topics that you would like to, or what are the experiments that you would like me to discuss about in greater detail. You can ask me right now also. You can also ask me in the discussion forum. You can also ask me in the next class. And I'll be sharing both the recording of today's class as well as the past year assignment questions in the with you in the Google. So there is a Google sheet that is there. I will be updating all of this in that. So does anyone have any specific questions? Okay, I take it that no one has any questions for today's class. In that case, I will stop the presentation right now, and I will be providing these materials to you in the Google Sheet. Thank you very much for attending the live interaction session. I look forward to your presence in the next class. And in the next class, I will also share some interesting experiments that were carried out regarding the week 3 lecture videos. I will share with you some interesting tidbits, some experiments, and so on. Thank you very much, everyone. I will now stop the session.
Thank you, ma'am.
Welcome.
Thank you, ma'am.
Welcome.